welcome to this summary video for the HIV resistant mutants lesson. This lesson is primarily all about genetics. That's really kind of the heart of the material that we're going to learn. You're going to learn about genetics in the context of a group of people that are resistant to HIV. So there aren't a lot of videos discussing the HIV-resistant mutant populations, but there are several good readings. So for this particular lesson, I really want you to take the time, make sure you take the time to read the background, the foundational readings that are listed. So you've got foundational readings. There are 3 of them. They're all short, but they do a good job of explaining one of the populations of people that have been found to be resistant to HIV, and that is a group of people who have a mutation in a receptor called their CCR5 receptor. And we did see that previously just a little bit, and it was described in a video in the HIV pathogenesis lecture or lesson. That CCR5 is one of the 2 receptors that are needed for HIV to enter into cells in the early phase of infection. Later on in infection, the CCR5 is replaced with the CXCR4 mutant frequently. But the CCR5 is really key for establishing the infection. And what has been discovered is there are a group of people out there who naturally have a mistake in their CCR5 receptor protein. It's a mistake that doesn't affect them health-wise, but it does because the viral specificity of the virus glycoproteins and the receptor is so specific. That tiny mistake, that mutation in the CCR5 protein has resulted in HIV not being able to enter into these cells. So these people can get exposed to HIV, and the cells that have a mutant receptor cannot be infected. So there's a group of people that are being studied, have been studied for a long time because of their repeated exposure to the HIV virus. And you know, their failure to get HIV. So read the foundational information on that. And that one of the interesting things that I think are described in those foundational documents is the idea of why this mutation exists. Because HIV hasn't become, you know, a human plague. It didn't become a human plague until really the 1980s. Although it was around before that, but it didn't turn into such a human issue until fairly recently in its history. But this CCR5 mutation has existed in the DNA long past, or previously, long before that. So why would this mutation be there? Why would it be helpful for people to have this mutation if the HIV virus wasn't a problem then? Well, there are a couple of theories, but one of the interesting theories is that the bacteria that causes plague, called Yersinia pestis, this bacteria, it looks like this bacteria uses CCR5 to do part of its damage. So if you had a CCR5 mutation way back in, you know, the Black Plague, right? Then maybe you didn't get infected, or you didn't get as infected, and you were able to survive. So the people that were left over after the plague went through and ravaged populations were the people who had this mutation, who then had kids who had the mutation, and the mutation got passed on. So you don't need to know great details about the plague clinically. I do want you to appreciate that it's still seen today. This is not an ancient disease that died out. Plague is still around. The Yersinia pestis bacteria can be found in Texas. There are cases of plague that happen, not large numbers, but they do pop up occasionally. There are 3 primary clinical manifestations of plague. There's pneumonic plague, which is a respiratory disease and is really, in most practical senses, the most contagious form because you can cough out the bacteria. And then there's bubonic plague and septicemic plague. Bubonic plague is characterized by these giant masses called buboes that are found around the lymph nodes closest to the entry site of the bacteria. So the bacteria comes into your body, it gets filtered through your lymphatics, and then you get this massive rush of inflammation at the lymphatics where the bacteria is gathered. The bubonic form very frequently turns into the septicemic form. And septicemia, remember, means organism in your blood. So in the septicemic form, the Yersinia pestis bacteria is in the bloodstream. So this, was a major, major killer before antibiotics. Now it is not nearly as big of a deal. That's why you don't hear about it much, even though it is still around. Treatment reduces the mortality of it dramatically. Okay, another interesting disease that comes up when you talk about the CCR5 mutation actually is smallpox. So smallpox is one of the famous infectious diseases of the world. Everybody knows that it's really bad, basically. Smallpox has a very high mortality rate, so a kill rate, very, very high, pretty close to about half of the people who get the variola major virus will die from it. We don't have much treatment to it. Fortunately, smallpox has been basically eradicated. I have to say basically because there are samples of it in freezers and research labs and things, but it's been eradicated from the human population. So hopefully it's not something you will ever see, and it will not come back and bother humans again. But those who study the variola viruses have found that that virus also uses CCR5. So this may have been a way for selecting for that mutation. People who had that mutation didn't get as sick from smallpox and were able to pass the mutation on to their children. So all of that being said, that brings us to kind of the meat of the lesson, and that is, what is a mutation? What does it mean? What is genetics? How does DNA work? How does RNA work? What is all this about? So most people understand that a mutation is some kind of mistake or change in a person. You think of like X-Men, that type of things. They're mutants. So that means they're different. But what does that actually mean in a scientific sense? So there's a series of videos by Amoeba Sisters that I really love that go over the basics of genetics and DNA and RNA and proteins and all of that good stuff. And then there's 2 videos by Crash Course Biology that go over the exact same thing. The Amoeba Sister videos are a little bit slower. They're a little bit more kind of fundamental. The Crash Course Biology is a little bit faster. It's a little bit more condensed, but the same basic info is there. So you can pick whichever series of videos you like if you have one preference over another, or depending on your background. Or you can watch both of them, which is what I always suggest if you have the time to do it. That set of videos is about half an hour for the Amoeba Sisters or the Crash Course Biology videos. So that will get you through the basics of genetics. And fortunately, bacterial genetics, prokaryotic genetics, and eukaryotic genetics are very similar at the fundamental level, at the basic level. There are details that are dramatically different when you really get into the higher level classes and really, really, really dig into the specifics of the processes. But for what you need to know for your career aspirations for this class, the basics, those are really the same. So if you've learned them in other classes, then this should all sound very, very familiar. There are only a couple of things that I do want to point out that are different enough, I think, that it's worth mentioning. One of those things is the fact that the DNA of prokaryotes are circles versus linear pieces of DNA in eukaryotes. And that creates this kind of interesting situation when you're replicating it, when you're doubling that DNA. For prokaryotes, there is one start signal called the origin of replication, and there is one stop signal called the termination of replication. So the process of copying the DNA will start at the origin, and the machinery will march along the DNA until it hits the termination signal, which is the stop signal. On a linear piece of DNA, it makes sense that you kind of go from one end to the other end. But when you're dealing with the big circle, it's important that you don't that you have a start and a stop, so you don't just sit there and stay in an infinite loop of running in circles and circles and circles. But it's interesting, the location of the origin and the termination, they are exactly opposite of each other on the circle. So what happens is actually a really cool, clever adaptation that prokaryotes have. What they do is they have two different sets of machinery, two different sets of helicases and polymerases and single-stranded binding proteins that will start back to back at the origin. And they will march down the DNA until they hit the termination. So you've got one set going one way, you've got one set going the other way, and then they smack together at the termination signal. And what happens there is the two pieces of DNA are hooked together. So basically, you've got a division of the work. You can get the entire circle copied in half the time because you've got two different groups that have split the work in half, okay? This is called bidirectionality. Bi is two, directions, bidirectionality. And eukaryotes don't have that luxury because they're linear as opposed to circular. The fact that the DNA has two strands is still the same. The setup with the leading strand versus the lagging strand is still the same for eukaryotes and prokaryotes. With that mention, I do want to emphasize that you do not need to know the lagging strand. You are not going to be responsible for DNA primase or ligase. You're not going to be responsible for the concept of Okazaki fragments. Of how that works. That's just not really necessary for your understanding of this process and the way that we're going to use it in this class. So all of that is presented in the videos, and if you learned it in other classes, awesome. But don't get caught up if that's something that is difficult for you. Don't waste the brain cells stressing out about that for this class. Okay, the other thing that I want to point out that is kind of unique to prokaryotic heredity is the way that information is brought into a cell. So, with eukaryotes, think of humans. If you're going to make a baby, you got the mother cell and the father cell. The zygote cells will fuse together to make the baby cell, da-da-da. Okay, so you got the mother DNA plus the father DNA equals baby. Well, bacteria, most of the time, are growing asexually. They're just copying themselves. The babies are identical to the parent. So what happens if that bacteria, that species needs to evolve? What happens if it needs to have new information? How does it do that? Well, it can't on its own. So it's got to have some way of picking up DNA, picking up information from the environment around it. There are actually three different common ways that we have found that bacteria will do that. So we've already alluded to one in the prokaryotic lesson, and that was conjugation, which is basically the equivalent of bacterial sex, where you have a pilus, which is like a hollow tube, that will form on one cell that will puncture the recipient cell, and then DNA can go back and forth through that tunnel, through that tube, and you can get a combination of DNA between those two cells that already exist. So it's not a baby cell being made, but it's the original cells kind of get remixed. So that's one way of getting new instructions to make things. Another way is transformation. Transformation is there's DNA in the environment and the bacteria picks it up. This usually happens when a bacteria in the environment dies. So one bacteria dies, they start falling apart, their cell wall, their cell membrane, everything breaks open and their DNA is exposed. And the other bacteria in the area can grab that DNA and bring it inside of themselves and see if there's any new information in that DNA that they don't previously have. And that might be a way of them getting new recipes to make new things. And the final way is transduction. So conjugation is sex through the pilus. Transformation is picking up free DNA from the environment, and transduction is DNA being moved from one cell to another through viruses. So this is particularly phages that will come in, insert the phage DNA into the cell, and then when they leave, sometimes they accidentally grab some of the cell's DNA, and then when they infect the next cell and they insert the DNA back in, they can accidentally carry DNA from one cell to the next. It sounds, I don't know, kind of technical, like sure, but does that really happen? It actually really does. It's been shown that viral DNA is spot all throughout different cell types, and evolutionarily it looks like this was a major way of moving information from one place to another. So that's how you pick up new information. How that information is used is really universal. You take that DNA are the instructions. DNA doesn't actually do anything, it's just the instructions on how to do things. So you take the DNA, you make a copy of it because the DNA has to last for the entire life of the cell. You don't want it to get ruined. So you make a small copy of the DNA, of just the section of the DNA that you need, just the instructions for what you're making that moment. You make a small copy of it, which is called RNA. And then that RNA only lives short term. So if it gets destroyed, then it's no big deal. You just go back to the DNA and make another RNA copy. So once that RNA copy is made, that's the thing that's actually read and actually, brought into the kitchen and cooked, if you will. So that RNA set of instructions is read and used to make the product, which is a protein. So DNA is kind of the cookbook. RNA is kind of the, the printout of the recipe that you actually bring into the kitchen and get dirty. And then the protein is actually the food that you would make after cooking. Okay, so you'll go through all of the details of, what DNA looks like, what RNA looks like, how DNA is copied, how RNA is made through a process. DNA copying is DNA replication. RNA being produced is a process called transcription. And then how that information is translated into the actual product, the protein, and that's through a process of translation. This lesson will also bring in the details of mutations, and you'll be able to see how a single change at the DNA level could result in the protein not being right. Think of if you took a recipe and switched the word sugar to salt. It doesn't matter what you do every time you use that recipe, it would never be the correct product because the recipe itself was wrong. That's what a mutation is. Okay? So it's just kind of a cool example, of the CCR5 mutation is a cool example of this in real life. There are many, many, many different diseases. There are many different benefits, like cool evolutionarily natural selection kind of benefits to mutations. We'll bump back into that concept in the antibiotic resistance lesson, how a single mistake can actually benefit the organism in certain situations, just like with the CCR5 mutation. If it keeps you from dying from the plague or keeps you from dying from smallpox, that accidental mistake is a wonderful thing, and will be a benefit for you, and then you will be able to multiply. And so the population, the genetics of that particular protein will shift in the population because you're the one who's able to have babies the most, and your information will be passed forward. So this is really a fundamentals of genetics lesson. Again, if you learned about it in cell biology, you learned about it in high school, then great. This should be a nice review. With this information, you can get an extraordinary detail. I mean, there's so much research done on this topic. You only need to know the level that is presented in the PowerPoints, the level that are presented in the videos. Don't feel like you need to have a PhD in gene expression at the end of this lesson. If you have any questions over this, as always, post it in the Muddiest Points discussion board, and myself or your peers will hopefully answer those questions for you. Good luck.